All right. This is not going as far as it did the other day. Oh, that's right, because I had my laptop. And here I am talking randomly. So you guys will have to speak up. So question 1, which type of systematics emphasizes common descent only? Cladistics, right? So why doesn't traditional evolutionary systematics only emphasize that? Why doesn't it?
I think it's more interested in morphological and physical similarities.
Yep, so definitely the morphological similarities in traditional evolutionary systematics is important. Okay. What are the 5 chordate hallmarks? Oops. Yeah.
Dorsal hollow nerve cord, notochord, pharyngeal gill slits, endostyle, and postanal tail.
Okay, so we've got dorsal hollow nerve cord, we've got pharyngeal gill slits, we've got notochord, endostyle, and postanal tail. All right, create a mnemonic for remembering the Linnaean ranks. Yeah,
Keep pond clean or Froggy gets sick.
what was that?
Keep pond clean or Froggy gets sick.
Okay, so keep pond clean or froggy gets sick. I like that one. Yeah.
Kingsley, please come over for Grace Spaghetti.
So Kingsley, please come over for
Great Spaghetti.
Gray Spaghetti. Who else has got one? Yeah, Shay.
Kings played chess on finely grained sand.
Ooh. Kings play chess on finely grained sand. I like all those. All right, so be able to write those out maybe from more general to more specific. Okay, what subphylum do tunicates belong to? So tunicates or sea squirts. Yeah, Urochordata, so subphylum Urochordata. What would Amphioxus be? What subphylum is that? Cephalochordata. And what is the subphylum of our visiting rooster? And us. Vertebrata, right? So we are subphylum Vertebrata, and so is he. Okay, so we've got this next question. What are the 2 major schools of taxonomy? So what were they again? So traditional systematics and cladistics. What does a homeobox gene control? Yeah.
Box genes that control like structure formation and like the central axis of your body.
Okay. Yeah. And were you going to add to that, Russell?
That's what I had, yeah.
Okay, yeah, so they're going to control what structures form where along that central axis. So when scientists play with those genes, they can make things like arms where antennae are supposed to go in fruit flies. What are the 3 defining characteristics of the subphylum Vertebrata? I love how he just camped himself out in front of Mary. I hope you don't mind.
No, it's nice to have company.
All right, who wants to take this one? Yeah.
Notochord, cranium, and pair of appendages.
Okay, notochord, cranium, and paired appendages. And when we get into the evolution of the vertebrate skull, what you guys are going to see is that the expanded cranium is very important, because what happens with the fish is it's actually a box within a box brain structure, basically. And the bones expanded from that inner structure to the outer structure, and that allows bigger brains. Okay, we already have list the hierarchical system of classification. How about when did animal life begin on Earth? 600 million years ago, roundabouts. All right. What process occurs when neural and sensory organs in the head are centralized? What do we call that?
Civilization.
Cephalization.
Anonymous dingoes on the move.
What?
Said anonymous dingo was on the move.
Yeah, I'm not sure who Anonymous Dingo is. When the action potential is at rest, what are the charges on the inside and the outside? What if we just say which one's positive and which one's negative?
Would it be negative on the inside?
Negative on the inside, positive on the outside. And depolarization is basically switching those charges.
Would, like, I know like the resting, like,
Mhm.
potential, like, membrane is -70.
Yep.
Like,
Somewhere around -70 is the resting membrane
But
potential.
we don't need to know what it, like, specifically would be.
No.
on the side.
I just want you guys to be able to describe describe what's happening in an action potential, what the major steps are.
Could you explain what depolarization is?
Okay, so depolarization is the switching of the resting membrane potential from a negative to a positive charge. So let me look up, I know I have the lecture somewhere. Where is it?
You said switching from negative to positive?
Mm-hmm. On the inside. Structural, functional, I guess it's. I go to materials by week. All right. So this is the resting membrane potential at around -70. Once a threshold of excitation has been met, then it's going to trigger an action potential to occur. And so the action potential occurs when sodium begins to enter the cell, and that's going to change the charge from a negative to positive. So this is depolarization when it goes up, and hyperpolarization when it goes down and exceeds the resting membrane potential. And this hyperpolarization occurs because of the extra potassium that leaks out at the end. So it's kind of a resetting process that over resets before it's ready again. So this might be a really good question. What's happening at steps 1 through 6, right? Do I need to wink, wink, nudge, nudge, say no more? Right, so know that the sodium channel's open, sodium begins to enter the cell, potassium channel opens, potassium leaves, and then basically the sodium channels become refractory, meaning that they close essentially and no more sodium gets into the cell. The potassium continues to leave the cell, which is going to reduce that membrane potential to the resting membrane potential, but as the potassium channels are closing, a little bit of extra potassium diffuses away, and that's why we get the hyperpolarization happening. All right, so how do synapses work? First of all, let's ask what the two types of synapses are. Yeah.
Like, there's, do you mean like there's one that goes from muscle and there's one that goes to nerves?
Those would be two different types for sure, but there's two major classes that are related to how the message gets from one side to the next. electrical and chemical. And so we looked at an example at the neuromuscular junction, right? And that one, was it electrical or chemical? It was chemical, right? And what neurotransmitters were involved in that one?
Calcium,
ACH, right?
The receptor and the sodium channel.
Yep. All right, so you have, this is how it works. An action potential comes in, and it's going to trigger the release of these ACH vesicles. And once those ACH molecules bind to their receptor on the membrane, that's going to allow the sodium to go into the muscle, and this is going to propagate that action potential over the surface of the muscle cell. And remember that muscles are capable of having their own special action potentials, right? Because they have a couple of different organelles that are specially for them. Do you guys remember what those might be? I know that's jumping ahead one lecture. Yeah, T tubules. And what else is involved? Uh-huh. So specifically those lateral sacs, right? Okay, let's see. Do dendrites or axons carry action potentials away from the soma?
The dendrites.
Want to try another guess?
The axon.
Yeah, because what dendrites do is they take the information into the soma, and then the axon basically carries that information out to its axon terminal, which is going to interface with other dendrites. What's the most common type of synapse in the human body? Is it an electrical or chemical? It is, yeah, chemical. We have a lot of neurotransmitters in our bodies to do a lot of different things. Describe the sodium-potassium pump. Here, I'll do this. All right, so here we have the inside of the cell and the outside of the cell. And the sodium-potassium pump uses ATP in order to move sodium out. Because if you notice, there's a lot of sodium in the outside of the cell, so it's moving it up its concentration gradient. And notice how there was a lot of potassium on the inside of the cell. So this is also going up its concentration gradient. So for every 3 sodium that are moved out to the outside of the cell, 2 potassium are going to be moved to the inside of the cell. So why would we have 2 ions going to the opposite of their regular concentration gradient? Because remember, if we talk about simple diffusion, we're going from high concentration to low concentration. So why might a cell require there to be a concentration gradient? Yeah.
It's like restoring after a action potential.
Yeah, so it allows it to restore after an action potential. And think of the potential energy that is available when you store some of these compounds such that there's a maximum gradient. where it goes from, you know, a very high level to almost nothing. It's like letting water go through a turbine in a dam. And so obviously those are very important in how action potentials work. So what's the difference between afferent and efferent neurons? Yeah.
Afferent is sensory, efferent is motor.
Yeah, that's a really nice way to describe them. So afferent is sensory and efferent is motor. So afferent literally means that those stimuli are being received by the central nervous system in some way, and then a motor response is what results. So that information is processed either in the spinal cord, or the more complex, responses are going to be integrated by the brain. But there's always going to be some sort of motor response. And for everyone who's not here, I brought my rooster into the classroom, and that's why we all keep laughing, because he's sitting right in front of Mary on the power outlet, preening and flapping his wings and blowing her notes away. Okay, what percent of cells are inner neurons? Yeah, Natalie.
99%.
So 99% are inner neurons. What are inner neurons? Yeah.
They connect the different neurons together to make a single pathway.
Okay, so they connect neurons together and make a single pathway. Is that what you're going to say, Russell?
Yeah, just they.
Yeah. All right, what does curare do to ACH receptors? Yeah.
It binds to them, and there can't be an action potential, so your muscles like stop working.
Yeah, so instead of the ACH binding to the ACH receptors, the curare does, and this is not the right message, so you're not going to be able to move because it's blocking the information necessary for an action potential to occur. What is a nerve impulse? Yeah, Russell.
Isn't it the nerves that are sent out both chemically and electrically?
Okay, so basically the nervous responses that happen, and we can say that it's an electrical impulse, right? And that electrical impulse can be connected through a synapse chemically as well. But if we were to give it one name that sort of summarized everything, what would we call it? An action potential, right? It's a very simple way to think of a nerve impulse. And does the way an action potential occur differ between myelinated and unmyelinated neurons? Okay. So in unmyelinated neurons or non-myelinated, how do they travel? They go the full length of the axon. Once you get that myelin sheath, do you guys remember what cell in particular wraps around the axon to form that myelin sheath? Schwann cells, right? The Schwann cells, they make the myelin sheath, and that myelin sheath has gaps in between it that allows for what type of action potential propagation? Saltatory conduction, right? So leapfrogging. And that sounds like it would be, kind of crazy, but it does skip some of the axon length, and it is able to move our nervous impulses a lot faster. So when people suffer from things like multiple sclerosis, what's happening is that they're actually losing their myelin sheath, and that's slowing down the way that they're supposed to be transmitting their nervous impulses. Okay, what term is used to describe the stiffening of muscles caused by a lack of ATP preventing myosin heads from releasing? Rigor mortis, right? So rigor mortis occurs because ATP is needed to reset that cross-bridge cycle. What term is used to refer to the area that acts as the functional unit of a muscle fiber. I'm sorry, Mary. I could come grab him if you want. I just think it's funny how he's like, oh, look, Mary, she's nice. Okay, so what is the functional unit of a muscle fiber? What's the smallest level that contains the machinery for the muscles to contract? Myofibrils. And then if we look even smaller within that myofibril, there's some other unit, the sarcomere, right? So microfibrils and sarcomeres, those are the very smallest functional units of muscles. What is the Z line? Here, go hang out with them for a while. All right, what's the Z line? Yeah.
It's like the boundary where actin molecules are attached.
Okay, so it's like a boundary between actin molecules. What two proteins are associated with the actin filaments? Yeah.
Myosin, one of them?
So myosin, when it does bind, it is related, but there's two in particular that are actually on the actin filament. And they're going to involve preventing that myosin head to bind.
The
Tropomyosin and troponin.
complex.
Yeah, so often they refer to it as the tropomyosin troponin complex. So how does actin and myosin react with and without ATP? Well, we already know what happens without ATP, but how is ATP used in order for a muscle contraction to occur? Yeah.
ATP allows for the myosin heads to be released from the actin filaments.
Yeah, so ATP allows the myosin head to release, and then what happens after that happens?
Sarcomere shortens, so the muscle.
Yeah, so you have the binding. Let me find that lecture for you guys so we can look at it.
Make a little bit of a mess of it.
That's his job. I'm gonna take you guys right to the cross bridge cycle. Here we go. All right. So you have the priming stage is when ATP binds to release that myosin head from the actin. In order for the binding to occur, the troponin and tropomyosin have to move away from the active site, and this is because of calcium. So when calcium attaches to the troponin, it's going to move it out of the way, allowing that myosin head to bind to the actin molecule. And then there's the power stroke that causes the movement of the myosin head along that actin filament. Once the movement has happened, ATP binds to allow that myosin head to release. So it's they call this cross bridge cycle. And if you notice here in this picture, you have the red structures here actually refer to the actin, and the myosin is kind of the darker structure. And so that whole sarcomere contracts because the myosin is actually moving along that actin. So there's actually a minimum and a maximum distance that they can either contract or they can lengthen. And that's limited by that. So just be able to describe this cycle and what's going on with it. Oh, this is the wrong one. So what are the four main steps of the sliding filament theory? I think we just covered that, right? Is actin a thick or thin filament? It's a thin filament. What dimensions change when a muscle is contracted? Bless you. What dimensions change? Has something to do with lines. Yeah.
Almost everything shortens.
Mm-hmm.
Except. Except the Z line?
Okay.
Something.
That's all right. You're on the right track. Okay, let's see. Yeah, here we go. The distance between Z lines is reduced because you have them sliding closer to one another because of the movement of those myosin heads. The width of the A band stays the same because you can't change the length of the myosin itself. Width of the I band is reduced because you have that myosin and actin overlapping there and causing a darkening of that area. And then the width of the H zone completely disappears. Okay, the H zone, that's the area that was kind of light in color. It comes from that German word Heil, and because everything's overlapping, it's blocking out the light. So just make sure that you guys know which of these dimensions are going to change. How does muscle fatigue occur? Yeah, Russell.
Muscles usually tire when they're stimulated with electricity.
Okay, so they tire when they're stimulated with electricity. What's the underlying cause, though? Yeah.
The calcium isn't like replaced fast enough.
Yeah, when you don't replace that calcium fast enough. What are two organelles in muscle cells? They're two specialized organelles.
The lateral sacs and the T-tubules.
The lateral sacs and the T-tubules in the sarcoplasmic reticulum, right? All right, moving on. Why did some Devonian fishes possess both gills and lungs?
Aren't they able to go in both land and water?
Yeah, it had to do with the quality of oxygen in the water, right? Because the Devonian was a time of drought. It was warm. Warm water doesn't hold as much oxygen. So just like Russell says, they needed to be able to take advantage of some of the oxygen in the water and in the air. This period in history is classified by drought and poor water conditions. The Devonian, right? What were the armor plating on fish or ancient fishes used for? Yeah, why would they have all that armor plating?
Is the obvious answer just protection?
Yeah, so they had to protect themselves against things like those giant Eurypterid water scorpions. Name one of the selective pressures observed during the evolution of jaws. Bless you. Yeah.
Like larger predators.
Larger predators, that's a good one. What else? A more complex nervous system. So larger predators and a more complex nervous system. What 5 groups of fish developed lungs and or swim bladders?
Does 5 groups mean like what classes or like,
The smallest taxa that we have.
so would that be subclass then?
It depends on what group we're looking at.
Okay.
Hmm?
Oh,
Yes.
okay, so I have to
Hmm.
class Placodermi.
So class placodermy. Hmm.
And then class Acanthodes.
Okay, and class Acanthodii. Okay, what else? Who else has them?
Subclass Dipnoid.
Subclass Dipnoi. Hmm. And like that's the most famous one, right?
Subclass Rhipidistia.
Yep, subclass Rhipidistia.
Chondrostide is the other subclass as well. Chondrostide.
Oh, Chondrichthyes? Hmm. Is that 5? I think that was 5. What class is closely related to bony fish but are now all extinct? Hmm? Yeah,
The placoderms.
placoderms. What is the largest group of vertebrates?
Is that the osteocytes?
Osteichthyes?
Yeah.
That's all right. Yeah, I know what you mean. Osteichthyes, right? The bony fish. What are 2 structures of Infraphylum Gnathostomata class Placodermi? 2 structures. Not really sure. The question's asking. Like 2 specialized structures, maybe? Can anyone think of anything? No? Yeah, I'm not really sure what the question's asking.
There's a PowerPoint slide. It says structures, haired appendages, and the second one is group of jaw vertebrates and predators with large eyes and mouth with teeth. I don't know if that's what.
Fragments. Those are strachoderms. Placoderms. Paired appendages. Large eyes and a mouth with teeth. That's probably what that was asking. Okay, we already talked about the Eurypterids. What subclass of fish includes lungfish again? Which one was that?
The
Dipnoi.
Dipnoid.
And we already know the environmental conditions in the Devonian. Sea lampreys are ectoparasites as adults, and their larvae are. Think of how Amphioxus gets its food. It's the same way as Ammocetes gets its food, right? Which is. Bingo, filter feeding. What physiological device allows sharks, rays, and skates to detect the electric fields of their prey's heart? It's got a funny name, kind of. It has Lorenzini in it, yeah, so ampullae of Lorenzini. This term describes fish that go upstream from the ocean to breed, so from the ocean to freshwater. What would that be? Anadromous, right? What if they went from fresh water to the ocean to breed? That would be catadromous. Define oviparous and viviparous. I mean, this comparison's really easy, right? So if oviparous would just be what?
They were eggs with a lot of yolk.
Yeah, they lay eggs with enough yolk that they can just hatch on their own. But viviparous species, if we define it generally, how do they give birth? Through an egg, or what do we see on the outside?
It's not just like
Just live birth. We just see live organisms coming out instead of being encapsulated in an egg. Okay, how do sharks stay afloat? Any Shark Week fanatics?
Is it the lateral line sensor?
The lateral line system allows them to detect some pretty complex changes in water, pressures and temperatures and things like that, but there's something that they're able to do physiologically that allows them to stay afloat.
Is it like through gas exchange?
They don't have gas exchange in a swim bladder. That's a good guess.
large livers with oil for buoyance.
Say that louder.
Large livers with oil for buoyance.
Yep. A large oily liver. What are the two lineages bony fish are separated into? So the two major groups underneath the osteichthyes. Uh-huh. Sarcopterygii.
Yeah, and then
Actinopterygii.
Yeah.
Those are terrible to say. Okay, how do fish respire? Are we pretty comfortable with how that happens? How the water goes across the gills in one direction and that they have a double directional countercurrent exchange happening with their blood that maximizes the gradient of oxygen exchange, and they can get a lot of oxygen out of water that way. So if you were to see a picture of countercurrent exchange or a picture of fish gills that were not labeled, would you be able to describe what you're seeing? There may be some images on this exam where I say what's going on in this picture. Okay, what is the difference between the lateral line and the ampullae of Lorenzini? I like that question. I like all these, but that one in particular is very interesting. Yeah.
The lateral line sensors are dealing with sensing like disturbances in the water, but the ampullae of Lorenzini are for detecting like the electrical fields of other organisms.
Yeah, that's exactly right what Mary said, which was the lateral line system detects disturbances in the water, and the ampullae of Lorenzini detects electrical fields. So I know we've gone a little bit over, and I've got to get Axel ready for his date with Science 101. So 7 o'clock p.m. on Sunday, that's when we're going to have our study session. We'll have it here, and we can go through the rest of these questions, and we'll go through all of the study questions, okay? And hopefully by then I'll have the exam written so I can give you a few more hints.